Hey everybody, for this last vodcast of Unit 2, we're going to explore movement via membrane proteins. So let's jump right in. Sometimes molecules need a little help, and here's where our handy-dandy, ever-so-trusted membrane proteins come into play. And proteins will utilize, basically substances will be moved via these proteins for a number of different reasons. Okay, maybe they're too big, maybe they're charged, and so we're going to need proteins to help transport these molecules from one side of the cell to the other. Now, we've talked a lot about proteins as far as structurally, we know their structure dictates their function. So as a result of this, proteins are very, and I mean very, specific, meaning they will only move a very particular molecule. So if I want to move one molecule, I need a particular type of protein. If I want to move another molecule, I need another protein. You can only imagine. So I'm sure it makes a whole lot of sense right now why we have as many proteins as we do. A form of protein-mediated transport is known as passive transport, or what we call facilitated diffusion. It's still, the principles of diffusion are still in effect. Movement of a substance from high concentration to low concentration, but in this case they can't just slip through the bilayer. They actually need to pass through a protein, and so here is where we get passive transport channels that will allow these substances to diffuse across. Ions are a great example of this because they can't just wiggle their way through, so these proteins are there to help them out. So here we have an idea of facilitated diffusion. More molecules outside the cell here. Okay, we need to get them in. So hey, protein binds on one end to the molecule. There's a shape change that sends that substance out into the inside of the cell. Here would be more of what it looks like on a little bit more of a sort of space-filling model with the cell membrane. Again, these protein channels are wedged in between the lipid bilayer. Glucose will move like this. It's a glucose transporter. We'll move them through. The protein shape will change to accommodate the molecule either going in one end and coming out the other. So this is just some examples. Because this is diffusion, no external energy need be applied, and it's particle or concentration driven. And again, it's diffusion. So the net movement will be from that area of high concentration to low, right down the concentration gradient. Sometimes, though, you need to move things against the concentration gradient. Sometimes you gotta go uphill. And, maybe there are times when there are, something in high concentration outside the cell that the cell just doesn't want. Enter this lovely process called active transport. Active transport, exactly as it sounds. When you're active, guess what you use more of? Energy. So active transport is energy dependent. We gotta burn a little energy to make it happen. Think about a boulder on the top of a hill. I can stand up there with the boulder. I can let it go. It's going to move right down that gradient downhill from high to low. I don't need to do anything extra for that boulder to go. Gravity does it for me. However, if I want to move the boulder up the hill, you best believe I got to be behind it and I got to be pushing it up that hill and hope I don't slip. Okay? But it's going to take some extra added energy from me to move that boulder uphill. This is the same way with active transport with molecules. It's going to take a little extra energy for me to move molecules from where there's a low concentration to high in the opposite direction or up the concentration gradient. And these do require special carrier proteins in order to make it happen. And they usually only move particular particles against the concentration gradient. Primo example is the sodium-potassium pump. Okay, this is where we will use one protein channel responsible for taking intracellular sodium. You can see boom, boom, boom for 3. ATP needs to get burnt, if you will, burnt, spent. So these bonds break, release energy in order to flip these molecules out. Without the added input of ATP, this molecule, the shape of the protein, the carrier protein doesn't change, sodium doesn't move. Okay, sodium potassium pump. Okay, so for the piece that involves the potassium, once the ATP has been utilized, okay, then we're going to bind potassium in on the other side and it's going to carry it across. So on one end, we're moving sodium out, and then once it's open on this end, potassium can come in and I can move the potassium into the cell. Other types of pumps, you've got calcium pumps, hydrogen ion pumps. You're going to see these pop up as we move our way through different parts of this year. And these are there because most often we have to be able to maintain a certain level of organization, a certain level of homeostasis, and sometimes that means maintaining particular concentrations inside a cell. All right, and to really wrap things up, another form of transport that a cell uses besides just moving individual molecules is what we call endo, exo, or pinocytosis. Okay? Endo, bringing things in to the cell. Cyto, cell. So bringing things into the cell. Exo, pushing things out of the cell. All right? And then there's pinocytosis. I'll get to that in a second. Okay? Here we have what happens in all of these. Okay? You have a vesicle that's made up of the same bilayer structure as the cell membrane. Whether I'm coming in, it doesn't matter. This could be in the cell. Okay, over here, let's say this is inside, this is outside. Okay, if that's the case, all right, what I have is that's going to attach here. Because it's made of the same lipid bilayer, when it attaches, it becomes part of the cell membrane and then pushes my substance out. Okay, in this case, this would be exocytosis. So this vesicle fusion with the membrane. This is to allow for a pretty significant bulk movement of materials. Cool. You ready? One more time. This allows for a certain amount of energy efficiency. If I can bulk move material out in one swift boom, then we have it. Okay. A little piece of information for you. I'm going to make you do a touch of work here. I want you to find out what this is. What is pinocytosis? I want you guys to take a look into that. Use your text, look it up online, come in for Whisk when we have our Whisk Disco with it ready to go as to what pinocytosis is. And can you give an example? So there we have it. We have completed Unit 2 as far as the content goes, and looking forward to all of the labs that we have to do to just enhance your knowledge of this stuff. So if you have any questions, don't hesitate, come talk to me, and have a great night. Catch you later.